Hello? Hello?
Yeah, hello.
Okay. Uh,
Can you hear me? Huh?
yes. Is the audience Bengali or mixed? Hello?
Sorry. Yeah. Yes.
Is the audience Bengali or mixed, or what's the proportion of the mix?
Audience is actually mixed audience.
Okay. Okay. All right.
So just we'll wait a few more minutes and then we'll start.
Yeah. Let me see. Share screen. Why is this moving? Are you seeing the presentation?
It's showing screen sharing, but we can't view your presentations. I think, ma'am, you need to allow your video first.
Oh, hold on 1 second.
Yeah.
You don't see the screen? Hello? Hello?
Yeah. Now I can see your screen.
Okay. You can see this, right?
Yeah. Yeah. Is it full screen?
No, that's what I'm trying to do.
Ah, okay.
Okay. Now you can see it?
Yeah, it's good now. Now all over the screen, no problem.
Okay. Gonna try to.
Okay, then just we will take a few more minutes because participants are coming in.
That's
Just
fine. That's fine.
just
I'm
five
just trying to.
minutes. Okay.
All right. And see this one. No, that's not right. No, it is. Okay, there's a little bit of a delay, it seems, with my mouse.
It
So
has gone to the second slide when you press it. Fine.
so now do you see the title slide or you see something else?
No, the title slide, Introduction to QBD, that one.
Okay. Yeah, I didn't have much chance to practice, so I'm kind of looking at this after completion.
It's okay, no problem.
So are
It's
most of
okay,
the
no problem.
attendees students, like have they graduated or are they, in the graduate level, PhD level, or mainly bachelor's level?
No, I think not bachelor's level. It would be more of postgraduate and mostly I think professionals,
Okay.
means either academic or industries, mostly professionals. And we are streaming it in YouTube as well at the same time.
Yeah, I saw that. It's
Yeah,
like,
yeah, yeah.
okay,
It's already
like
already
I can't say anything bad, right?
right,
Because
right.
it's
It
not, I
will
can't
be
like swear
it
or
will
anything.
yeah, it will be on the record.
Yeah, that's a problem.
Yeah, true. True.
I thought you guys were gonna post it to YouTube later. I didn't realize it was gonna be live.
It's live. Actually, the thing happened, what license we have procured from Zoom, it allows only 100 participant at a time
Right,
to
right.
enter. So
Mm.
parallelly we are running that YouTube live also, because if some more participants coming in, they can join in the Zoom. They can watch it over the YouTube. And at the same time, it will be recorded also in YouTube, so it will be there.
Okay. Are you able to do polls on your meeting to see, you know, how many people are in product development versus analytical development versus marketing and things like that, or no?
No, because for this we need to pre-work, means we should have asked for some form registration, something like this.
Okay.
Then they can mention that we can keep a track. And
Okay.
now we can ask in the chat box, but it will be random answering. So maybe we can get some idea. We can ask in the chat box.
Yeah, I think with my full screen though, I probably won't be able to see it. I don't
Yeah,
know
yeah,
where they'll show up.
yeah. 1 minute, ma'am.
Mhm.
Somebody calling. Yeah. Okay, I think we can move on. It's 6:08. Still participants are coming in, but let them join. We because we have 1 hour, including some question answer or interaction session. Okay, so am I audible?
Yes, I can hear you.
Okay. Okay, good evening, everybody. On behalf of JU Pharma Alumni Group, I, Bapaditya Chatterjee, wish to welcome you all to today's talk by Dr. Paramita Sarkar. Dr. Paramita Sarkar, definitely she is also a Jadavpur University alumni. She did her B.Pharm from Jadavpur University in the year of 1991. Later on, she did her PhD from University of Minnesota Department of Pharmaceutical Science, and after that she started working with industries. She is a pharmaceutical formulation scientist with more than 15 years of experience in different pharmaceutical, biotech, medical device, and consumer healthcare product development. And in her career, she worked for various big multinational pharmaceuticals and healthcare companies such as Pfizer, Pharmacia, Boston Lom, Mylan, and with a wide experience of working in regulatory field product development for generic liquid, semi-solid product development, and technology transfers. She worked as a leader of tech transfer for several years. She has also wide experience on formulation and process development expertise for sterile injectables, topical solution, non-sterile formulations, including different liquid dosage forms, different gel-based dosage form, novel drug delivery systems, and definitely on QBD, quality by design, and its application for formulation and process development. So today, Dr. Parmita Sarkar will be talking on Introduction to QBD in Pharmaceutical Product Development, which is a quite relevant and interesting topic, especially for industries and like us who are working in the professional field is increasing day by day in research oriented work in product development inside the lab. So I hope it could be an interesting one. And after the talk, for the participants, if you have any questions, any query, you can type in the chat box or question answer box in the Zoom. And those who are watching over the YouTube, please type your query in the YouTube chat options. We'll be happy to place it to our speaker. So without further delay, I would happily invite Dr. Paramita Sarkar, our very own, our university senior Paramita Di, to deliver her talk. So it's over to you, Paramita Di. Please, you can proceed.
Thank you, Bapadito. Can everybody hear me okay? Just give me an yes, Bapadito, if my sound is okay.
Yeah, I
Everything's
can hear you clearly.
okay. Namaskar, everyone. Thank you for the opportunity to let me share a presentation on quality by design. It's something that is very near and dear to my heart through the years of pharmaceutical product development in the industry. And I was very happy when this process, this thought process became more formalized and adopted into the pharmaceutical product development mind frame across the industry. My apologies in advance if the presentation appears to be too basic, and however, we can definitely correct that by having more involved discussions on some of the points that I will touch on. So feel free to note down your questions as I go through the slide. Usually I like to be a little bit more prepared in that I prepare to go through the presentation without referring to the slides, looking at them. Did not have enough time for that, so if I'm taking a little bit of a pause to take a view of the slide that I put together before I speak to it, that's the gap that you will hear on your end. And I'll try to wrap it up so that we have enough time for questions and answers, because this type of a topic really is more of an interactive learning experience from people across the board, rather than just presenting some slides with some bullet points on them. So with that said, of course, the main thing is, what is QBD, or quality by design? And it's a systematic framework and approach that helps us define objectives and stress that during a product development life cycle, we are going to try our best to really understand the product and all the factors that affect it, in conjunction with the processes that are used to make the product that will ultimately enter the marketplace. And it requires an understanding of process control based on science and quality risk management. The main objective for adopting this QBD mind frame is based on the fact that quality really needs to be built into the product at every stage of the product life cycle, rather than tested into it. And you will find me repeating this on a couple of different slides, simply because it is a very important component of QBD and risk management. And it is a good idea to kind of have it as a mantra at the back of your mind when you are evaluating your pharmaceutical development program. So as I mentioned before, QBD is a continuous and iterative process, and it can encompass any sphere of your pharmaceutical development life cycle. We're going to focus mainly on the product development that encompasses the formulation development, the process development, and touch a little bit on analytical development. It is absolutely possible to dedicate full sessions to each of these three individually. So the discussion really is going to kind of touch the surface of all of these, so that the interactions between these various spheres that you're seeing over here is a little bit clearer. The underlying goal to bring a product to market, you always have to have the patient in mind. If the drug isn't safe, and if the drug isn't effective, and you haven't proven it to be safe and effective, it's not something that the patient really needs, and it shouldn't be in the marketplace. Drug product development is continuous, and the QBD principles and guidelines are enshrined in ICH Q8. Again, a product cannot and should not be tested into compliance. This is something that many of us who are in the pharmaceutical field, starting out early, and really want to impress upon their teams and team members in being efficient in developing a product, forget that just setting certain specifications and having tests that can test to those specifications is not the end goal of your pharmaceutical product, right? You have to understand where the concept of the product came from and where you're going with it. So there are, of course, different stages in a product development life cycle. Once in the preclinical evaluation, other than the drug substance synthesis optimization and target selections, um, that part of the QBD is something I won't be speaking to. We're going to be talking about the part that really gets us into making a product in a GMP environment and what's associated in the tech transfer phase of a product. And that typically the tech transfer, which is the handover from the development or the research and development side to the commercial manufacturing side. And here the QBD principles absolutely are essential to follow, because if you have a faulty tech transfer and the people on the other side don't understand what you're handing over to them, what's going to happen is the manufacturing side, the commercial site, is going to run into issues, investigations, deviations, and a lot of unexplained occurrences, which is not going to look good for a company. As I mentioned, you can find the details of the QDB concept in ICH Q8, and it is intricately related to what is laid out in ICH Q9 for the quality risk management and the pharmaceutical quality management systems in Q10. So QBD, it has to be able to address the expectations that will come from and are incorporated into the Q9 and Q10 guidelines as well. I've included some links for sources of the information. Some of the information, I have cut and pasted information from slide presentations that are available online. So links to those presentations are also included. There is a wealth of information online, and you can very easily get flooded with the information. It's good to know what you're looking for and how much information you're looking for when you jump on the net. Otherwise, you will go down rabbit holes, and all of it is very interesting, but may not be applicable to the tasks that you are assigned to complete at that moment in time. So as I mentioned, the Q8, Q9, and Q10 principles and guidelines really work together. Starts with formulation activities, goes into process development activities, and then manufacturing, scale up activities, post-marketing surveillance, continual verification, and process improvements. For this presentation, we are going to be more focused on the first two aspects of the formulation development and process development activities. We'll touch a bit on how these will impact what happens on the manufacturing site, especially with regards to out of specification type scenarios and investigations, deviations, those type of things. So the background for your quality by design is that it has to be science and risk-based. And the QBD guidelines introduced the concept of design space and flexible regulatory approaches, and the concepts of quality by design through some specific examples. So when you read through the documents and the Q&A associated with the QBD ICH QA guidelines on the FDA website, there's a lot of clarity provided around what can be matrix, what can be bracketed, what can be used in the design space to make decisions easier on whether something needs to have a regulatory filing or not. And we'll clarify some of those in the upcoming slides. um, uh, just wanted to take a little break and make sure that everybody is hearing okay. And up till this point, there are no questions. Okay. So this is a schematic representation of a visualization of how the QBD approach could look like during the product development. And it's directing to a linear flow where you get the product profile, you develop your CQAs and do a risk assessment and then define your design space and control strategies and then look towards continual improvement. I'm going to take a little bit of a modification of this flow because what it doesn't show is the feedback loop at each stage of the process. So it looks linear in how it's laid out, but in your continuous improvement efforts in manufacturing, you may be able to provide feedback to the team that originally developed the product and transferred the product in for improvements and evaluations that will improve the efficiency of your manufacturing processes or minimize errors. And they can do a regulatory assessment with their regulatory experts on how that's going to affect the filing. So this is an iterative and continual process and stays embedded in the product development life cycle all the way until a product is taken off the market. So we're going to focus a little bit on the concept of design space and what that is. So when you start with a quality target product profile or QTPP, which I hope at this time is fairly consistent in all companies. Now, having said that myself, I have walked into an organization that understood that QTPP needed to be generated. So they generated a paperwork, but they didn't know how to use it. And they did it kind of in a vacuum. So I'm talking from a perspective when I was brought on to lead the tech transfer team at a manufacturing site. And we'd get this big dossier of documents from R&D, and they would have their critical quality attributes, QTPP, everything, with no back and forth with the manufacturing site on how they develop those specifications or those attributes that they want to hit. Now, I will give the allowance that because it was a generic product development, they may have felt that a lot of that is already defined by the RLD that they are benchmarking against. It didn't need as much involvement of the tech transfer team as you would need in, say, a new product introduction type of scenario. But that's not the case. And the reason is, while you have a framework when you're doing an ANDA type of product development and you're benchmarking against an existing product, you don't know exactly who their raw material suppliers are. Have they added any additional incoming specifications to their raw material to maintain their product quality? Have they discovered any particular raw material behaviors on storage that have led them to optimization of their product and their processes? So these are still unknown to you, even when you're doing reverse engineering and figuring out the QQ match for a generic product. So you need to keep that in mind. You will get some information, but you will not get information about the history of things that went into the product and process development and their learnings. And your manufacturing process can look very, very different than what the innovator product uses. So you have to be mindful of that, and your QBD still has to be applied in a methodical manner from the beginning once your QQ match has been done. So you narrow the space for the composition. That means you know the list of ingredients that you want, and you know the levels of ingredient that you want. But you don't know the order of addition. You can kind of guess at it based on the solubilities, the type of finished product that you're going to get. You also don't know yet what the impact of lot-to-lot variability of your raw materials is. So are the critical material attributes as defined by your raw material specifications as it comes from the manufacturer, is that sufficient? Or are you going to have to develop some additional specifications? Sometimes it could be particle size, sometimes it can be microbial load, depending on the risk and the type of product you're putting it in. And you will have to work that into your design space and specification setting processes. Now, all of that is fine and good, but if you don't have robust analytical methods that can distinguish between small or significant changes in your formulation, then you're not going to be able to design a space, right? You need to have the right parameters that you're testing against, and those parameters have to be indicative of performance, safety, as well as shelf life. So if your test methods are not robust enough, that there's a high degree of variability or a wide range of acceptance, then your design space exercise is kind of going to be futile, and that needs to be remembered. One of the things that was highlighted in the question answer sections of the QBD document is that for once you have designed space for your formulation optimization, and I'll have a slide later on that gives a visualization of design space. And you have small variabilities in the material attributes within that design space that requires you to adjust a level for whatever reason. Typically, that will not need a submission or regulatory post-approval change. And this is very important because it can save you a lot of time and effort in going back and running through the entire process where you're getting your internal team together to do an evaluation. Then you're presenting it to the agency, waiting for the feedback before you can implement changes that are possibly an improvement to your product and process by making minor tweaks within the design space. So that is definitely a plus point in having a design space. Having said that, there is no requirement on having a design space for a pharmaceutical product. It's a recommendation. It's an adoption of best practices, and it is ideal if the tech transfer SOPs or best practices document within an organization address the concept. However, it's not something that the FDA is going to look at your pharmaceutical development portion of it and go, where have you defined the design space? This is very different. I don't know how many of you are involved in devices. In the device world, however, there is a concept of the device history filed, and they will look at and ask for how you reached certain decisions and decided that the device is going to perform in a certain way all the time. So there's a little bit of a expectation different whether you're in the drug space versus the device space. Are there any questions for design space for a product? Okay. Now, once you've understood your composition variations, interactions between ingredients, and boundaries, which is your design space for the product, you're also going to have to look at how that ties into the design space for the process. And this is going to be dictated by, and this is where the QTPP, it cannot be a document that is created in a vacuum by the R&D team and then just put in a pharmaceutical development file and thrown over to the manufacturing sites going, hey, catch, I think I've done all my work and you guys have to make it work. The QTPP really needs to take into account what is going to be your year one or launch volume, what is going to be your year-on-year growth, and what do those volumes look like? Because the market, the manufacturing site is going to be scratching their heads like, okay, I've got the ability to make 100 kg batches and 1500 kg batches. They're kind of out of the 10 factor level, but now you want me to make something in the 1000 kg or 800 kg batches. It's not an easy jump to make it from the 100 to the 800, and it's not an easy jump to make it from the 800 to the 1500, regardless of how well you've said that your manufacturing vessel, your mixing equipment configuration are the same. There are absolute differences in what happens in scale up, even if the principles of operation and design of your equipment remain the same. And that is something that is oversimplified in the tech transfer process, where it's assumed as long as you're within this arbitrary 10-factor scale, you're at low risk. And I would absolutely stress, when you're looking at commercialization and an aggressive timeline, that you consider these things up front, because once you put in certain batch sizes within your regulatory filing, if you go out of them and you haven't done a design space, you're looking at additional filing, additional validation, okay? For packaging presentation, why is this important? Now, a lot of the times people go, oh, I'm gonna put it in a plastic bottle. But what if the manufacturing site goes, okay, I only have one plastic bottle and it can only fill up to 15 milliliters. So regardless of whether you have a 5 mil, 7.5 mil, or a 10 mil fill, I'm gonna give it to you in a 15 milliliter bottle. Well, the thing is, has the R&D team definitely looked at the impact of the package presentation on the shelf life of the product to come back to the manufacturing team and make a case for, no, I need a 5 milliliter bottle with a 4.5 milliliter fill because I'm going to try to minimize headspace, or I'm going to make sure that the surface to volume ratio is not too much. All of these need to be considered. So make sure that the impact of the packaging presentation takes into account the facility and equipment requirements, as well as the product shelf life expectations. And this has to be evaluated on the pharmaceutical development side. Also, in terms of defining the process parameter impacts on the critical quality attributes, everything from your mixing, your hold time, your filtration step prior to filling, all of these will have an effect on your product performance and shelf life. My background is largely in liquids and semi-solid formulations. assuming that many of you are working in, of course, the most widely used method of dosing, which is oral solid dosage forms. So my ability to speak to the variabilities in those type of processes is going to be a little bit more limited, but maybe during the question and answer phase, if you have anything specific, other people that are tuning into the presentation will be able to contribute to the discussion. The advantage of having a very well thought out design space for your manufacturing process is simple. When you get into validation, for folks who are familiar with the old traditional mindset of validation, you needed to run it at the extremes of the process to show during process validation that it works. And sometimes everything did, and sometimes you didn't, and you didn't understand why things failed, and you were redoing your validation, right? So if you take the effort and put in the time and money to have a very well thought out design space for the process, that's some of the anxiety and heartburn that you can avoid down the line. And you can do your process validation within that space. And you can also make certain adjustments to your process within that space without affecting your validation process. So this again needs to be explored at an earlier phase in the development studies. And it is really a joint effort between the scientists in the lab scale who are able to observe things much more closely, even when they're making things at a 5 kg or a 10 kg level. A lot of the things are very manually adjusted. They can take the cover off of something and observe it. When you get to larger and larger scales, those type of interventions and observations become more problematic. They compromise the sterility of the product or the continuous processing of the product, so may not always be possible. So it would be when the pharmaceutical development team gives a process flow diagram to the tech transfer team or the manufacturing team on this is what we're recommending. The best thing to do is to have some of the engineers observe the lab batches and see what the scientists are seeing when they are making some decisions or conclusions on what a process needs to look like. And I'll give you an example. We were doing a formulation for a gel, and in the lab, we were very happy having, 2 or 3 different beakers of pre-mixed components that then we would combine slowly in the final phase. And then we do our little magic and do our pH adjustment in the final step. And voila, our beaker had the beautiful finished product, and we tested it and everything looked good. The engineer comes into the lab and observes us doing it, and we had very well written steps and lab compounding procedures that we follow. And he said, this needs to be a 1 vessel process. It's a sterile product. We are not doing that many transfers. You are putting it as risk, and we're definitely not doing a pH adjustment at the end. So what we thought worked great and we had used to do some of our scientific stability formulations, the engineer came and said, no, that's not going to work. It's great, you've done 2 kg and 5 kg batches, and you've provided things for your GLP studies and your small scale and some of your phase 1 studies, but this is not going to be a commercial product, you need to come over to the manufacturing site and we'll show you the smallest vessel that we have and what we do for products that require a pH to be buffered within a certain range. You have to really pinpoint the composition of the buffer that's going to give you that pH at the end and optimize the process accordingly. So I'm going to take a little break again over here and see if there are any questions or I need to go any slower.
No, but I think fine. You can go in that pace.
Okay.
Questions are coming in. Maybe I'll take it later on together. Is it okay?
Sure. Sure.
Okay. Please proceed.
So in addition to your scientific understanding of your materials that go into your product, as well as the equipment, the process parameters, you also need to apply a statistical approach in getting a design space. And one of the most common approaches that you will see, of course, is the utilization of design of experiments approach. And it is a multivariate analysis that provides a more efficient approach to understanding the design space. For example, if somebody were looking at a design of experiments approach to running a chemical synthesis, they would have input parameters or factors such as temperature, raw materials, and concentration, and to assess at the same time to figure out the conditions in which a response or the output can be optimized, such as yield, impurity level, etc. And we'll look at an example of such a design space. The design of experiments studies usually requires fewer experimental repetitions. And I want to stress, not fewer experiments, it's fewer experimental repetitions. What that means is when you're doing the one factor at a time or n-1 experiment where you're varying this one and seeing if things change, then varying this one and seeing if this change, and then you're looking and you hit on both of those experiments certain thresholds beyond which the formulation of the product is behaving very, very strange. You don't know whether that was a result of just going over, or was there some sort of synergistic result in component 1 and component B within the previous concentration ranges that allowed for a certain behavior to occur. So now you can't narrow down where along the variability spectrum you need to optimize, okay? It does provide a better process understanding in less time and shorter development cycles in manufacturing. We'll also look at some of the barriers to adopting DOE on a regular basis. Sometimes it has to do with the fact that you don't have access to a dedicated statistician, the understanding or application of the DOE and utilizing certain programs to generate a DOE design of experiments format just isn't there. And again, you will be doing more experiments, but you will be spending less time figuring out where the optimization is. And so in doing a design of experiments exercise, it is important to understand that the factors of interest are varied systematically from a lowest end to a highest possible value, and all possible factor combinations are executed in the same set of experiments. The design of experiments approach can be applied in the formulation screening phase, as well as for the final optimization. The difference would be in the formulation screening phase, your output would be more simplistic, like yes, it works, no, it doesn't. So just two outputs. Whereas in your optimization, you would try to narrow the yes response that you get into a range of yes, the pH is between 5 and 10 to the pH is not above 10 to the pH is between 5 and 7. So that type of parameter differences will come up in screening versus optimization. So just a pictorial representation of what the design space can look like. You see that this one is looking at 3 different parameters at 2 levels, and a high level and a low level. And if you were to do the full set of experiments, you would end up with 8 individual experiments, and you would measure the responses. Over here, they have shown the responses as the product yield and the impurity level, and they chose one that gives you a high enough yield and a low enough impurity level for the optimization for that process. It is very important when considering doing a design of experiments exercise that the factors should represent the parameters that have the strongest influence on the experimental outcomes. So in most cases, your formulation product or process is going to have way more than 2 or 3 factors. But you need to be able to have a good enough understanding to choose the 2 or 3 factors that you think are going to be the most important in determining your outcomes. Otherwise, you're going to end up with a lot of unnecessary experimentation and information from those experiments that are really not going to help you in building quality into your product, because you just have missed the key critical aspects of your formulation or your process. So that again requires cross-functional team engagement and discussions before you embark on a DOE exercise. So get all your team members involved, at least the experts in each of those areas, whether it is the scientists who develop the formulation and the engineers who are going to be heading up the scale up and manufacture, and the analytical scientists who are going to be testing it. Everybody needs to be involved in coming up with an appropriate DOE exercise. Once you have a design space defined, and that's in, used for the basis of your decisions and your planning, remember that a design space that is not well thought out has limitations, because you cannot extrapolate. You can't say, well, my data was trending in a certain direction as I varied the components in an upward direction. If you haven't measured the response within the design space that was evaluated, you cannot make that extrapolation. So what can lead to an afterthought of quality versus having it built into your process and your product? The lack of processes and analytical robustness, processes that are kind of static. So you do this step and then you don't look at the effect on the next step. It's not contiguous. You don't look at changes that can happen over time in the process. High variability in your measurement systems, when you don't understand or characterize your raw materials. And I really want to stress this one This is where a lot of a headache comes in once a product is manufactured, because um, because of the time constraints, the development team will say, well, we want these specifications, raw materials. The sourcing team will go out and get it. Sometimes it's a high priced material, but the R&D loves it because it's really good. When it comes to manufacturing, that really pushes costs out. And manufacturing then decides to request an alternate source with the C of A. But what maybe the R&D team didn't specify to the tech transfer team is every time that we've gotten this product from somewhere else, even though it matches the C of A or the monograph, we found a level of, say, peroxides that was not acceptable over time. So when you get the raw material in, it'll pass all specifications, but the R&D has the information on what that level of peroxide needs to be able to give you a stable product, but that hasn't been communicated to the tech transfer team. So the characterization of the raw material is really, really important. You need to understand lot to lot variability. That needs to be done at development, not after it's transferred over. And that needs to be also addressed through getting different manufacturer samples. Always have a primary source and a secondary source for your raw materials qualified. Otherwise, you may run into a situation, this has happened, where we were getting compendium material from a supplier, the only supplier that we qualified for that particular raw material. The monograph specifications were updated, which required us, from a regulatory perspective, to update our specifications. Now, when we bought the raw material, we knew that the raw material wasn't designated as NF. However, it was all, the certificate of analysis always used to say it will test to NF. So when we said, okay, you need to also update your specifications to comply with the monograph, they said, no, we don't. That's going to be more work. And we supply to various other companies. They haven't asked for this. So we were in a conundrum because once we've updated our internal specifications, we're kind of not following good practices in being able to accept raw material from a vendor based on established specifications. Also, when your test processes are not well understood, you will have frequent out of specification values. And a lot of the times, and you'll see in some examples of the 483s that I've highlighted later, a lot of it is brushed on the carpet as operator error or lack of training or something like that. The FDA hates to see that. They absolutely don't like that. If they see too many of that, they're going to tell you that you don't know what the heck you're doing and you don't have a very robust analytical methods development, validation, and analytical methods transfer program. Again, data trend isolations among functions. What does that mean? That means the QC lab that's probably doing finished product testing. And in some cases, you will have a separation between the QC lab that does finished product testing and the personnel that do raw material testing, and then personnel that do the stability program of it, and of course, people that do the manufacturing. They will not communicate things that are looking out of the ordinary in their own silos, or any training that they see, and it won't be done on a timely manner. What that means is that the information remains siloed and the connections that need to be made to appropriately react in time without this situation getting out of hand really goes to a critical state. Another thing is the lack of knowledge management and management of the current expectations. What does that mean? It means that the documentation, the information management, how that is compiled, how that is reviewed, how that is transferred over, how that's trained on, and how that is evaluated on a periodic basis is really not well defined or well managed. So people may have really great SOPs that say, hey, we do this for our product development, we write our beautiful pharmaceutical development report, we write our beautiful tech transfer report, and they go and write their beautiful validation report. Nobody really cross-checks between them to look at the information. Do they really jive with each other? And is there a feedback mechanism to bring your development team back into the process when situations arise? And it's very important to have those demarcated in your process, because the manufacturing team can go a long way in having minor investigations and deviations and dealing with them on a piecemeal basis without ever calling in the R&D team. And by the time they do, too many things have been done on a piecemeal corrective measure to really figure out what went wrong. So this, the whole pharmaceutical development process doesn't stop when it goes into manufacturing. And we must remember the quality of the product has to be designed into the product. Just because things are testing okay to specifications at the end doesn't mean that you will be able to address any investigations, deviations that occur, say, on stability. Because a lot of the time you'll see initial specifications passed. On stability, it's looking different. Now you're going, well, let's go back to the manufacturing record. Now your manufacturing record was written in such a way that even though ranges were defined, the team was trained to do basically their exercise always at fixed values. So there's no variability that was captured in the manufacturing process over time. I think I'm going over time, and there's about, I think, 6 to 7 more slides, well, 10 more slides left. So let me know if we need to break off and just go into question and answers, or we have time to go through the remaining slides.
No, ma'am, you can continue another 15-20 minutes. We can continue.
Okay. And sorry that I went over time. It looked pretty short when I looked at the presentation.
It's okay. Another 15 minutes will be very fine for us. No problem.
Okay. So understanding all aspects of QBD is critical for the proper risk assessment, right? Sometimes a familiar claim or assertion that's made during the product risk assessment is that none of the drug quality attributes are critical, or the quality or the critical processing parameters are not critical because the R&D team has chosen very specific material attributes and have chosen very specific process parameters. And as long as you stay within those, everything should be fine. And the FDA is, well, you haven't really explored everything. You haven't explored the limited, you're limited in your characterization and the sources of your variability. So you can't make that blanket statement. And I've personally actually had to deal in my tech transfer experience where when they did the R&D did the regulatory submission, they went from their initial evaluation of a process as everything high to everything low. And I said, you can't make everything low because there are things in the process parameters that link to your critical quality attributes. So it's great that we've defined the space around it, but it's still critical to your process. And if there's any scale up work and movement to other equipment that needs to be done, that needs to be highlighted as the critical part of your process. Otherwise, it will be missed. So we'll quickly go through some 483 examples. I'm not going to read through them line by line, just an overview of what they look at. And a lot of the time it focuses on the analytical methods because that's the most well documented part of your process, right? The QC lab is swamped in documentation, or should be. But you will get out of specifications investigations, and on, like I said, doing stability testing, and there'll be a hypothesis, possibly because there was insufficient sonication during the sample preparation. So that's your hypothesis. You need to have an experimental plan on how you plan to address it, and you need to execute all the experiments in your plan to justify that hypothesis. In this particular case, the agency goes, well, we weren't really sure how you obtained the low assay value when you hypothesized that it was due to a sonication problem. And yet, before you could really come to a valid conclusion, these batches were already released in the US market. So closing out investigations quickly is not desirable. What will happen is if the FDA keeps drilling and they find out that you didn't address the situation properly and you have product on the market and the out of specifications really could not be validated based on the hypothesis and the testing that you provided, you may be looking at a recall because you're not able to justify that the impurities, for example, that came up on your results are not a risk to the patient. In case of in-process control and testing, a lot of the, as I mentioned, will come as no assignable cause or manual error without documentation of the manual error. What did the operator do? And how was that determined as an error based on either a specific batch record instruction step or a specific SOP? If you can't define that and you're just saying the operator was incompetent, then you have another issue. So even when you say that it's manual error, you need to understand at what point of the process the error occurred, and that is done by interviewing the operators that were involved in compounding the batch individually and really walking through the steps and seeing if they understand the SOPs and the batch instructions. In my personal experience, here are some of the examples that I ran across. So the R&D team developed a viscosity specification which did not correlate with changes in a topical cream physical appearance and behavior over time. Why is that important? So, you know, in the guidance for the evaluation of semi-solid products, there is a homogeneity that you have to do. You cut open the tube and you look at the top, middle, bottom sections of your tube. In that homogeneity, it is not just an assay test. You're looking at the consistency and the appearance of the cream. So if there's phase separation, if there's formation of crystals, those also go into your physical appearance and visual homogeneity that has to conform. There was a viscosity test, which was a 1-point test. at a certain RPM that did not describe the flow behavior of the cream under temperature and stress conditions. So it's a parameter that we tested, that we validated, but it didn't mean anything for the product. In another case, the process parameter, a narrow temperature range was said, this is the most critical part of the process. But when we did the actual engineering batches and decided to explore the space around it, because it was very hard to maintain that narrow temperature range during manufacturing process. And from a manufacturing site, you don't want to be continuously doing investigations for going half a degree over a temperature range. And we were able to prove that that wasn't critical, but because that was already in the filing, we didn't want to do the extra work. So unfortunately, that parameter, at least until the time I was there, was not updated. I don't know if they went back because we started to get a lot of deviations from the manufacturing process in some of the later lots. A particular mixing step was wrongly identified as critical, and we kept on trying to optimize that step, whereas the later homogenization step was the one that was important in terms of the speed, the duration, and the temperature. So we were going down a wrong path when we were trying to optimize the process at the manufacturing site and trying to develop a validation plan. There was an example where R&D established specifications for particle size for a raw material, but it didn't correlate with what we were seeing in the finished product. And they have had completely different methods of evaluating the particle size, which the knowledge of how those could be tied together was not transferred to our quality control lab. So those are just some personal experiences. So what are some of the barriers to successful adoption of a quality by design? There is still a significant number of companies that have not implemented or are reluctant. They include, not clarity on the guidance and from regulatory agencies. So this is sometime in the past. Now the guidances are fairly clear. There are a lot of case studies that have been documented. One of the common ones is there's no process or quality advantage to be gained, because until you do the design of experiments, you don't see it, right? All you see is, oh my God, I'm going to have to do 10 more experiments before I can do my tech transfer. No, my management is not going to like it. And then a lack of the understanding of the basic quality by design and understanding where the return of investment comes from when you are chasing down and rejecting batches simply because you cannot nail down an underlying reason for failures or deviations. So the QBD approach has to be applied across the product and process development lifecycle, and cross-functional teams need to be involved at every step of the process. And it is in a continuous and iterative process. It has to be built into the culture. It has to be built into your best practices. And in your tech transfer documents, it really should be spelled out as a requirement. And at these different stages of product transfer and product development, it is up to the tech transfer team, even if they're going to look unpopular, to be able to step up and say, I'm not comfortable with this selection or these criteria or these analytical methods. I need to show how your specification justifications were set and what it may mean for me in the manufacturing environment. So again, the key takeaways is that QBD implementation requires a company quality culture. It comes from individuals that are passionate about it. It is an iterative process and a continuous process. It requires a cross-functional discipline, information and knowledge sharing, and it requires a good understanding of the science behind your products and your processes. And it utilizing things such as your DOE will improve your risk assessment capabilities, which is something the agency does look at. How are you doing your quality risk management? What is your approach to that? Application of quality by design results in more robust products and processes, and down the line, that means less risk for product failures and recalls. So I think that was it for my presentation. I guess we can do the question answers.
Okay. Thank you, ma'am, for your nice presentation. We can understand that QBD is a vast topic, but the way how you presented the overall aspects and how the basic features can be incorporated in the product development, that are really good. And especially, I like the last parts where you have mentioned some real examples of not complying with QBD or the causes of 483s, those
Yes.
things. So we
Yeah, the
have,
real life
uh,
scenarios are the most important to understand. These do result in real problems.
yeah. So just we have few questions before going into the participants' questions. I have one questions for you, just to understand that in the last slide you have mentioned that there is a mis wrong concept that it is costlier
Correct.
before the this one. Yeah, before this one, you have mentioned that it there's a wrong concept. It is costlier. So, doing a QBD during product development, as per my idea, I'm not this QBD is not actually my cup of tea, but just what based on my understanding, making QBD doing QBD during the product development could be costlier than the conventional one, but in long term it reduces the risk of product failure. Am I correct?
Correct. So a lot of your cost analysis for a product is based on what? Your time of entry in the market, and during that time frame, what other products are going to appear on the horizon, right? That's how your marketing and commercial team are looking at how quickly how you need to get on the market. However, once you get on the market, if you start having failures, you still don't have a product. So it's very important to stress that fact that you can skip some of the steps and go into your tech transfer with a very rudimentary understanding of your product and your formulation. But can you imagine holding up release of your batch once your launch is completed? Because now you're all of a sudden you're starting to see problems and you haven't had enough time to figure out why. That's a much costlier risk than spending a few extra months. Now, I absolutely understand because I have had these tussles with management, right? Where I was insisting for R&D to go back to the board, and I said I'm not going to allow you to do the tech transfer. And very few people will take that much of a hard stand because that makes you very unpopular with the scientists as well as the management. But remember, if as long as you keep in mind you're doing this for your company and to prevent any major recalls, and that is what's driving your decision, and you have to explain to them why. You have to be able to explain to that why you feel that there's a lack of understanding or lack of information.
Okay, then I'm going gradually to the participant questions. There is a question from some participants. Name is not written here. A little big. I'm trying to read step by step. The first part is he or she is asking, do you need to define the IPCQS linking it to the CQS? You need
Um,
to. Yeah. Yes, please, ma'am.
So yes, because if you're doing in-process testing, and you're setting specifications on that, and you don't understand how they relate to your final CQAs, I would say better not set those specifications. Uh, and here's what happens is you'll get, sometimes you will do the in-process testing, but because of limitations on how long you can hold your product and this and that, uh, you will go on to completing the product at risk. So you complete the entire compounding, filling, and packaging at risk while you're still waiting for either the in-process test, if there was a discrepancy, for that investigation to conclude. So now your entire product that has cost associated with its compounding, with its testing, with its packaging is just sitting there, and you're trying to, and you're also doing your finished product release, and that looks fine. So you are at a dilemma on what do you do with this fact. So do not set in-process controls and specifications unless you understand how it relates to your end product CQAs.
Yeah, very specific answer, ma'am. Thank you. Second part is you need to define, link your CPPs and link them to your CQS, right?
Yes.
Yes. Okay. And then it continues like this. Also, some parameters like line speed or compression speed needs to be validated in the outer limits of the design space, is what I understand. Increasing compression RPM beyond validated range, just citing that it's within the design space. Is that enough? Am I
I'm not
able
sure
to?
I fully understand that question. So if suppose you design a design space where you're supposed to do say 18 or 20 experiments, but 3 or 4 of those experiments um, don't work because the limits that you've set are beyond either your measuring capability or the capability of the equipment. So those experiments are not valid. So your decision can only be made on the data that's generated within valid experimentation values. I don't know
Okay.
if that answers the question.
Yeah, I also guess so. Here she is asking that increasing compression speed beyond a validated range, just citing that it's within the design spec. Is that enough, or it requires again a small validation or stability and just annually reportable? Means something from regulatory aspect is asking.
Um, okay, so when you did your design space, you had values that your validation program used values within the design space, did not reach the limits of that. So if your space was designed correctly, the theoretical answer is yes, but you would still need to do an engineering study to document that and do the tie-in. Typically, what the agency is trying to promote, and I don't know, um, I don't have enough examples of showing where this has been successfully adopted and implemented on a large scale. I'll look for some examples, but theoretically, that's what the agency is saying. If your design space is well designed and well tested with good scientific methods that are robust, you should be able to do minor variations within that space. For example, we did mixing ranges over 10 minutes to 90 minutes or 120 minutes. we did our validation between 30 and 90, leaving enough room should there ever be any deviations outside these ranges. We already have the data to not go back and go into a full-fledged investigation, but to show that, yes, we're okay. So it helps in your investigations and deviations as well.
Okay. Yeah, the next question is from Avijit Lambe. He's asking that at what stage QBD can be done and which will be the good design for QBD? I think the stage you have answered already, but again, it's up to you. The question is at what stage QBD can be done and which will be the good design for QBD?
Okay, so philosophically, QBD should be done at every stage of your pharmaceutical development process because you have variables galore, right? You're always looking at different things that are affecting bioavailability, your stability, when you're looking at drug substance, you're looking at yields and impurities. So QBD applies across the board. When it makes sense, really, is when you have enough scientific understanding of what you're dealing with. So if you are in the process of selecting, say, viscosity enhancing agents in a product, and you go right off the bat and say, I'm going to try and nail my HPMC concentration within 1% and 2%. You've kind of ruled out your exploration of other agents. You've ruled out a wider range, which can result in lower costs or improved mixing by using lower concentrations and yet getting same viscosity results in the end. So you need to understand, and this is where the QTPP is important. What are we designing? What do we need to achieve in terms of your release specifications, your stability specifications, your marketing presentations, your supply chain requirements, whether cold chain supply will be acceptable or not? All of that needs to be understood. So with a good QTPP, you can start working on what you're going to incorporate in your quality by design exercise.
Okay. The next question is from Mr. Pradeep Majumdar, probably your senior or batchmate, I don't know, but definitely a Jadavpur University senior. He has written that first he has thanked you, and then he has written that I work in early drug discovery and development phase. Would you please let us know the time to introduce QBD in early drug discovery phase? Lead selection or lead optimization at IND level, considering resource and time.
Sure. So I am a little removed from the early drug discovery phase at this point in my career. But early on, when you're doing the solubility studies, so again, when you're developing a product, do the drug discovery people know what the requirements is? So is it going to be a tablet, a capsule, advanced release dosage form, a multi-particulate dosage form, a semi-solid, a injectable? So you need to know that, right? Based on that, you will need to know, well, what volume does it need to be in? So can I, if the dose is very little, do I say you have to drink a gallon of this medicine to get your 2 milligrams dosage? What's your delivery vehicle? What's your delivery volume? So these discussions, again, need to happen and need to be described in the QTPP. Now the QTPP, what I want to stress is an evolving document until you get to the tech transfer phase. At that point, it really needs to be solidified. I'm not saying it can't be changed, but it needs to be fairly solidified at the tech transfer phase. At that point, if somebody comes back and says, I really wanted a red tablet instead of a white pill and I wanted blue writing on it, that becomes hard because now the formulation team is going to look at the pigments that need to go in, whether there's any migration during compaction of those pigments, whether those bleach or bleed out in any way. So then you're back to the drawing board. So in the drug discovery phase, you need to understand the route of delivery, the maximum size of your delivery vehicle, and your dosing. Is it going to be QID, BID? And in case of things, say injectables, what's going to be your site of injection? Because you're going to be limited by the tolerability of the excipients that you select in your screening studies.
Okay. The next question is from Sinas. Is asking when can we extrapolate the outcome from DOE data? If the design used was full factorial, how reliable is the extrapolation statistically?
So the recommendation is you do not extrapolate outside your design space. You can't. That is the recommendation. Now you're saying full factorial. It still just means you did all the experiments within the design space. You can do partial factorial because you have a limitation of resources and still get enough information with regards to the interaction of the parameters. But you really shouldn't extrapolate. That what extrapolation always means you're going outside defined boundaries, right? And unless you have, I would say don't do it, because any mathematical model, any response surface that is an outcome of your design of experiments is really not amenable to extrapolation.
Okay. Thank you, ma'am. The next question is again from Mr. Pradeep Majumdar. He's asking that if a small molecule and a device, if the development is for a product where small molecule and device are together as a single product, would you recommend to implement QBD separately for molecule and device or both as a single product?
That's a complicated question. So when you're having a drug device combination, and I touched on this briefly, you are going to be held to the CFR guidances on devices in terms of design history file. Now, of course, the key portion of your drug device is your stability and your dose delivery. That's going to tie into your molecule. And so you need to be able to do them. You cannot do them separately because they tie into each other. But you will need a separate design history file documenting your development of that device.
Okay, yeah. The next question is from YouTube chat box. I can't extract the name. The question is, if we are developing a novel molecule, how to determine QTTP and CQS?
So that's a good discussion. So when a program is started, it started with we need a new molecule to address this particular disease area, correct? And your QTPP is going to look a little bit different. What is it going to tell you? It's going to tell you we need the molecule to be have a greater half-life so that there's less frequent dosing, because the precursor to this molecule has shown that in this class, there's limited solubility and it has to be injected. So you have some information based on which you're embarking on your new molecule discovery or design. What are you optimizing? Are you optimizing for solubility? Are you optimizing for targeting? Are you optimizing to have less, say, protein binding, because that's what's limiting your bioavailability? So that your QTPP will look a little bit different, and you should know what your route of administration is going to be. That's going to be a key factor in optimizing your molecule and what your dose requirements are going to be.
Okay, thank you, ma'am. The next question is, I'll take a couple of questions. It's okay, huh? Two, three more. Yeah.
Okay.
Okay, so the next one is, how can you implement QBD in formulation and evaluation of fixed dose combination product?
A fixed drug combination
Fixed
products.
dose FDC.
Right. So you would start with the historical information you have for each of those. Now, if your company is looking at the fixed dose combination and was not involved in the individual development of the formulations of the drugs that are available, then what happened is all you've done is in your design space, you've introduced an additional variable, right? So you're not only varying the concentration of one API, you're varying the concentration of another API. So right there, you've increased the number of parameters that you're going to evaluate in your DOE. And in that particular case, I would say you absolutely should do DOEs, because the one thing that the one factor or n-1 experiments doesn't do at all is tease out interactions between components. So you may be at the borderline of the solubility of one component in your formulation matrix and doing very well on the solubility of the other API in the same matrix. And until you do the DOE, you will not know that failure point.
Yeah. Okay, next question is from Dr. Bapi Goren. He is asking, how shall we decide the suitable design of experiments during formulation development? Which DOE we should choose? How we can decide?
So you always want to narrow down the parameters that you're exploring. When you're doing your formulation, you have to take a two-step approach. You're going to do a screening DOE, where your criterion isn't a measurable response, it's more of a pass-fail. So does the formulation pass antimicrobial efficacy testing? Does the formulation have a pH above 7? Those kind of things. So it's going to be a yes or no response, and that'll help you simplify your experimentation. Then once you've done that, you can select a few of the key ones that look like they're having an effect on your formulation space and choose that for your optimization DOE. So you have to do DOE in 2 tiers.
Yeah. Okay, ma'am. My last question is from some YouTube viewer. During DOE plan, how to determine at what level parameters need to be studied? I mean, the factors need to be studied.
That's going to be based on your scientific understanding and also the capabilities of your process. So if you set a requirement in your DOE that your impurities need to be below, say, 0.001%, and yet your detection methods really can't go that low, that's not a very good rationale for that design, correct? So it needs to be based on what effect you're trying to get and what your measuring capability is of that effect
Yeah.
and how reliable it is.
Okay, thank you for your very specific answer. And I guess that's all from the participants. Some suggestions are also coming. Somebody is telling that it will be better if you can explain the concept with relevant case studies or illustration. But I guess that it needs some type of preparations, and we can come back maybe some other day for some real case studies or some real illustrations from your side. Then it will be
Yeah,
much
sure,
better.
sure. Yeah, I have a whole different presentation just on one case study for a DOE. So DOE can get very involved, and that's a topic in and of itself. It's shown as a subset and a good approach in your quality by design framework, but that is a topic in and of itself. Even tech transfer, the actual case studies for various tech transfer from the examples that I gave, those are topics in and of themselves that'll take up an entire hour.
Yeah, maybe some other day we'll sit together to learn that one from you, because that will be more practical to understand how to go with, especially for academic researchers. We me I'm working in academics, and I know more few of the participants are here are also from academics. So we need to understand how it is practically applicable, because from our part we mostly use only the DOE. But what I understand, DOE is a tool to design the space, to reach the design space. Am I correct, ma'am?
Yes.
Yeah. So then from DOE to go to overall QBD from an academic perspective, we need to learn maybe some other day in future. You'll have some other sessions. If you feel free, I can arrange another one for real case studies.
Okay, maybe a couple of months from now again. We'll
Yeah, yeah,
talk.
definitely from your convenient schedule. Okay, so with that, thank you very much, Paramita Di, for your nice presentations, and especially very good interactive sessions. A lot of questions, and you have answered those questions very well. We have got a lot of inputs, lot of takeaway messages from your presentations. So, with this, anything you want to convey to us beyond this, Didi?
No, thank you, Bapadito. And I think Bedoduti also got me introduced to
Yeah,
the
yeah.
group. So thank you for arranging my talk and letting me speak to an audience. Of course, you know, Jadavpur University family is always like family. So feel free to reach out with any questions. You guys have a great WhatsApp group going. So I check messages. I don't always reply in the chats, but I always check what's going on. So
Okay.
stay connected, learn from each other, and don't be afraid to challenge conventional thought processes and adopt new practices to make the products better. Namaskar.
Okay, thank you. Just to share with you, you can also find in the chat box some of your seniors. Devasis Chakraborty, Head India Operations of Piramal, is here with us, and then Pradipda is here with us, Bedoditi, and so many seniors and some juniors, even some undergraduate student is also here from
Great.
UG2, some researchers from CSIR, IICB, and some faculties from Malaysia, some definitely from India. Abhijit sir, Dr. Abhijit Mojumdar is also, I don't know, maybe your contemporary in Jadavpur
Oh,
University.
yes. Yeah. Obijit is my junior, my
Oh,
guy.
okay. He is also here with us. So it was a nice session indeed. Thank you, ma'am. Thank you very much. We will definitely sit back with you in near future with the case studies. Okay.
Okay. Thank you. Namaskar.
Okay.